Rubins, and I'm a NASA astronaut. I'm also a major in the Army Reserve.
And let's start at the beginning. So I understand that you were born in Connecticut and then raised in California. Could you talk about how where you were born and where you grew up influenced your life?
Yeah, so I grew up in the San Francisco Bay Area in Northern California. And some of the kind of scientific influences in my life were all of the museums there. So my parents were really interested in making sure that the kids had good scientific exposure. And so we went to a lot of science museums. And one in particular, the Exploratorium in San Francisco, that was a big hit amongst the kids. And so we went there a lot. My dad took us there one day, and there was actually a conference going on at the Exploratorium. This was a scientific conference, like a real conference, with academics that were presenting their work. And, my dad's kind of a funny character. He said, let's go to this conference. This looks neat. I don't know if it was just a way to entertain the kids on a rainy Saturday afternoon. But he registered us for this conference, and we went, and I got a chance to sit there and listen to scientists give their presentations, give their research talks. And I think I understood maybe 10 or 20% of it. I was 12. I hadn't had any kind of molecular biology education, but it was just so interesting to me. And so I just remember I sat there and I listened to them the entire rest of the day. And it was a conference on recombinant DNA. That was really big back then. And so I just remember thinking, this is so amazing. I want to do this when I grow up. And so I do think that kind of this environment, where I grew up in Northern California, and just the fact that my parents took me to these museums and really encouraged that exploration, and it's not really specific to that region, right? Because there's science museums everywhere, but that was pretty impactful.
Were your parents scientists?
No, they were not. They're social workers, therapists, ministers, nothing related to science. My grandmother was a laboratory technician at Cold Spring Harbor Labs. And so I did hear a little bit growing up about her working in the labs. Back in the days, she had to wear heels and nylons to work every day. But I did think it was this, it seemed normal to have a family member that had worked in the lab.
Did she tell you stories about what it
Yeah,
was like?
yeah. She told me stories about Barbara McClintock and jumping jeans, and I heard about corn of different colors, just like kind of growing up as a kid.
Oh my gosh, that's so exciting. And what were some of your early pop culture influences during that time? Were there books that you read or films that influenced you?
Gosh, in high school, not so much. I do remember when I got to college, and this is probably of my generation, there's probably a whole bunch of people that would say this, but Gattaca was a movie that I absolutely loved. And it's kind of funny coming full circle now. I have a career in space and did a lot of genomics work. So I thought that movie was pretty, and it stands up well to time. If you go watch that again, I think it's actually got a lot of really, interesting issues that are still at the forefront even today.
No, we talk about Gattaca a lot here, and how to prevent Gattaca-like futures in many
Absolutely.
ways. Very important.
Yeah. I thought they did a nice job on the science as well. And, having something like that in the pop culture, at least it brings awareness of genome sequencing and like what the ethical implications might be.
Do you feel like you were the Uma Thurman character in that movie?
Oh,
Or
I don't know who I was. I just enjoyed the whole movie.
who are some of your early mentors, in addition to your grandmother and your parents?
So I went to UC San Diego, and I had been working on HIV. I got into HIV work because my dad, who worked for the public health department, there was a program there where teenagers would do peer-to-peer education on HIV to prevent transmission and infections. And so in order to make sure that we were really giving the accurate information, it was a year-long program, and we had several months where we met weekly. They brought in scientists, they brought in clinicians, and they brought in patients. And so that was incredibly impactful at that program to be able to both listen to the patients who in the late 90s, it was still a struggle. Integrase inhibitors hadn't been invented yet. We had protease inhibitors, but there was resistance that was happening. And so it really seemed like this area of research that we're getting there, but, and there's a lot of promise, but this virus just keeps evolving and causing resistance. And so I became very interested in that. And I think it was really neat to have that interaction of both the clinical side, the research side, as well as the patient side, and to have all of that in one setting. So it was an incredible program. And then we went out to schools. We came up with a kind of a play, a presentation. We went out to schools. We did the play for them, and then sat and answered questions. The idea being is like maybe you're going to really connect with somebody that's your own age, but make sure that they're educated in terms of the information that they're giving out. So that was like a pretty good mentorship program, and really drove my interest in virology, and looking at HIV and looking at how the virus mutates, what's going on with the viral sequence, and how that, why is it able to get around all of these drugs? That was a big question for me. So going into college, I like, number one, I needed to earn money, and number two, I wanted to get a job in the lab. So I really, thinking about it now, I'm like, what was I thinking? I just kind of cold called the Salk Institute and sent my resume in. And I said, I'm very interested in virology and HIV. I'd like to work in the infectious disease lab at the Salk Institute, as like a 18-year-old. So I sent my resume in, and they actually, I think since I had some HIV work on there, they gave me an interview. And so I worked with Dr. Rick Bushman at the Salk Institute, and that was my very first lab job. And of course, when you have no lab experience, you got to start out with things like autoclaving and making solutions and making sure you can measure things right. And so I did that and kind of worked my way up to actually running assays. And so Rick he was an incredible mentor. Thinking about it, when I was running my own lab, I always try to make sure I had a lot of undergrads in there. But people come in and they've got skill sets that are ready to be developed, but they don't necessarily come in. You've got to start somewhere. And so undergrad students, I found them amazing. I loved having them in my lab, but they take a lot of work to get them into the place where they can actually be productive and do research. And so I found it very fun, but it takes a lot of time. And so I kind of think back on this and I'm like, wow, like I really appreciate what Rick and what the rest of the infectious disease lab did for me. Taking on an undergrad student had, just cold called the Salk Institute that I want to work there, and teaching me what it is to work in the lab. Because I did that even before I was in any laboratory classes through my degree program at UC San Diego. And so that was my first experience with science. It was very profoundly impactful. And I think I got a lot of freedom in the lab to talk to the postdocs and to figure out what they were doing with their research. I started running assays on HIV and how that integrates into the genome. And like a good way back when story, these assays were run on polyacrylamide gels between glass plates with radioactive materials so that we could sit there and we counted the ladder. And I'm kind of glad that I got a chance to participate, because I was like, I feel like that's like the legit old school way of doing sequencing all the way through, to this modern, nanopore and single cell sequencing that we're doing today. Like I feel really good being one of the, just at the end of the old guard, doing it the very hard way.
No, you really proved your mettle back then, I think.
It just took a long time, it took a long time to get, whatever 100 base pairs that you were looking at that day.
So working on HIV in the 90s, I know that was still a time where there wasn't a lot known necessarily, and there was still a lot of education that needed to be done with society. Was it the biology that really attracted you to that area of research, or was some of the social science also of interest to you?
Yeah, I think it was both. I really came in from the biology because, so at 12, I said I want to study DNA. Like that was a given. And then kind of coming into this group and learning about, okay, well, here's this virus and it actually integrates into the genome. And, this is a tough social issue. And then I think the social piece really came from the patients that we were speaking with, some of whom, they were quite sick, and hearing their stories and thinking, being able to directly correlate the scientific research you could do could have an impact on this person's health. I think that was very powerful as a young person to experience that up close and try to, draw that line from what we do in the lab to how that's actually going to help people in their everyday lives.
And at some point, you pivoted more to cancer biology and went into the Ph.D. program at
Yeah.
Stanford.
So I worked in the Salk Institute, the infectious disease lab, and then about a year later, I also got a second job. I really needed money to pay for college. I got a second job in a lab that was doing cancer diagnostics. And so this was at Althea Technologies. And so we were designing PCR assays to look at, essentially where you've got two chromosomes that are coming together and you've got, that's resulting in a cancer. And so we were looking across that junction to define what had happened there and use this to diagnose at a molecular level what was going on with this cancer. And again, it was very forward thinking in the late 90s, and so I really liked that. So when I was applying to graduate school, I applied to both the microbiology and immunology and cancer biology programs, and I was kind of trying to figure out which way I was going to go. And so I went to Stanford for graduate school, and I went to the cancer biology program, and it was great. It's the several years of coursework, and I really loved it. But when I did my lab rotations, I started out. I did my very first rotation in Pat Brown's laboratory, who'd done a lot of work with microarrays and cancer, and so that made sense. And I knew, like, from week one, I was like, I really want to work in this lab. I had to go do some other rotations, but I knew I was coming back there. And the cancer work was incredibly exciting. But he said, look, I've got this opportunity. I know you come from an HIV background. We have some new infectious disease research that we're pioneering, along with David Relman's lab at Stanford, and then also Peter Jarling he was with the Army Medical Research Institute of Infectious Disease. He said, are you interested in working on this project? And so I started doing that, and it was very clear. I was like, I definitely want to spend the next 5 or 6 years of my life working on the interface of genomics and infectious disease.
Can you talk more about what it was that really made you so passionate about it? Or like, what was it that it really appealed to you about the science work that you were doing, or like about the field of science that really excited you at the time?
That's hard to like pinpoint what was incredibly exciting. When I was a kid, like when I was 5, they said, people, oh, what do you want to do? And I said, well, I'm going to be an astronaut and a biologist and a geologist. Because I assumed, like, of course, you can have 3 careers simultaneously. And the astronaut thing slipped away in high school. Some of it was people were like, well, you need to figure out what you're majoring in college. Like, you can't major in astronaut. Like, what? Also, this is a job that not a lot of people have. It's not very realistic. So you need to at least have a solid plan B. And of course, science was always at the forefront of my mind. I think it was just this idea of discovering something. When I was 12 and I was at this conference and I'm watching people present their discoveries, and the idea that they did not know this yesterday, and they go in the lab and they can figure something out, and then they can come up with a story and they can understand how something works. So this idea of understanding how DNA works just blew my mind as a 12-year-old. And it just lodged in there really deeply. And so then, when you put that together with, well, now we want to understand how a virus works, how that something so small, just little bits of code, could be so devastating to a human. But then the fact that we've got all of this scientific expertise and we can actually, for the first time in history, overcome these kinds of things. And I think that was it was this idea that, like, you can go discover stuff every day. You get in the lab and your job is to discover things. And people always ask, like, oh, did you want to go to med school? Not in the least. I absolutely appreciate that there are amazing physicians and nurses and clinicians in the world. It was never something that I was drawn to, and I would make a horrible doctor. I think it was not, I didn't have that kind of side of the art of medicine. But I just wanted to be at the lab bench. That, it was just very clear that I wanted to be a researcher.
And you still have time to get your PhD in geology.
The funniest thing is that at NASA, because we're returning to the moon and we're collecting rocks on the lunar surface, I've had a lot of geology training now. Yeah, I'm not going to get a PhD, but I do get to talk to geologists all the time. And I absolutely love it. And I just put in a scientific abstract to do DNA sequencing on the lunar surface to try to identify what pathogens we might bring with us into outer space. So yeah, I'm mixing it like I got that blend. I'm mixing it all up.
Your 12-year-old self was
It's
absolutely
pretty
right.
exciting. I think most scientists, when they get into the lab, they sort of do feel like a 12-year-old. Everybody just gets really giddy about their work. They get excited about what they're doing.
And at some point, you also went into some other infectious disease areas, as I understand it, working on Ebola and other very scary sounding sorts of viruses. Did you work in like, you must have been in like very high like biosecurity sorts of environments? Was that good training for eventually being on the space station? What was working in those sorts of environments like for you? And was that during your grad school career?
Yes, it was during grad school. And so this project that got presented to me by Pat and by David Relman and Pete Jarling was, let's try to look at gene expression changes during the course of monkeypox or smallpox infection. And again, this was, the human genome got published while I was in grad school. And so we were using microarrays to look at this. And we were looking at levels of gene expression on the human side to try to understand what was happening either in human cells or in our animal model. And so that was kind of my introduction to the poxvirus world. And I would say I really, if you asked me, I would say I am a pox virologist. That was kind of the virus that I concentrated on the most. And then, when you get to know the smallpox people, you meet the Ebola people, and it's a tight-knit world. So I started doing some Ebola and Lassa fever and Marburg research as well. And that any of those viruses have to be researched in a biosafety level 4. So that's the highest level of containment that we have. It's these incredibly secure facilities. We wear a full body suit that's got its own air source. So there's some big hoses that come in from the ceiling, and that pressurizes the suit, and it actually circulates the air inside the suit. And then the air comes out the back, so that you're always getting fresh air, which is a completely separate source than the laboratory. And that's how we make sure that the virus doesn't enter the suit, that the human can still breathe in there. But yeah, it's people walking around in these big white spacesuits. They're clunky, they're cumbersome. It's hard to do work when you sit down at a laminar flow hood and you try to put your arms in, the suit sort of puffs up here. So I have this habit, if I sit down at a laminar flow hood, I always sort of push my clothing down because I'm used to like having to take that suit and tuck it under just so that it won't puff up into my field of view. But that kind of really awkward, difficult way of doing science with these huge rubber gloves on was my norm. That's what I did for my graduate school career. So when I came to NASA, the idea that you would put on this giant cumbersome spacesuit that makes work really difficult, it wasn't that much of a transition for me. And I do have to say the spacesuit is a lot harder to work in than a biosafety level 4 suit. But it was part of what I grew up with in the scientific world, about working with something that's very dangerous, rigidly following the safety protocols, kind of understanding the impact that an accident in that area could have, and paying attention to detail and really understanding what the safety responses are, and how to appropriately respond to them. That was very early in my graduate career. That was definitely ingrained as a part of it.
One thing we try to talk about here at NIH is how important failure is in science, and how, especially earlier in your career, you have to understand how to work through failures, experimentally and theoretically. What sort of failures did you struggle with when you were in school and in your early career?
Everybody's got their story of like the thing that went wrong in their PhD. We had one particularly challenging, thorny problem, which was we were doing microarrays, and so these are little like laboratory slides with a bunch of dots of DNA on them, and you put your sample on it. And if you've got a matching bit of RNA or DNA that we've converted RNA to DNA, it's actually going to bind to the slide, and it's got a fluorescent molecule in it. So then it's going to fluoresce, and so you can look at all these dots and you can identify, what gene expression is there. So for us, that meant how are the immune cells responding to the virus? We can actually see what genes they're turning on or turning off by seeing if those transcripts are present. But it's got a fluorophore and it's on a slide, and you put it in a little laser scanner, and it's one of the dyes is red and one of the dyes is green. And so you come up with these scans, and we would do, hundreds of these. All of a sudden, we just started having these problems where it looked like this hazy red background glow across all of the slides. And you're looking at a ratio of the red to green. So if the background is red, it messes everything up. And it takes quite a while to process these. So you spend several days processing them, and you put them in the scanner, and all of a sudden everything has had this like red glow of death. And we hated it, but it would come and go. So like some days we would have it, some days we wouldn't have it. And so of course everybody goes and tests their reagents first thing. They make sure we got the right batch of things. We look at all the chemicals. We did a deep dive into like every chemical solution in the lab. We looked at our water system. We looked at the laser on the scanner. We couldn't figure this out. And we finally, in some conversation with people and came across other people that were having these issues, somebody said it was due to the ozone fluctuations in the atmosphere, which was not in our list of things that we were looking for. And so when we started to drill down into that, we're at Stanford, we're in the San Francisco Bay Area, based on sort of the cars and the pollution and whether or not it was a bad ozone day or not, we would have this red glow and this red haze on our slides. But this process took months. It was a lot of frustration. And so it was this kind of like failure of experiments that we learned from, and it was incredibly interesting. And once we had that cause, we could do things like build an entire environmental box. So my lab bench in grad school turned into like this hermetically sealed box where we would scrub ozone in order to process the samples. And then it's like very hard to do the bench work. So then I started partnering with a robotics company, and they built a robot that could do all of this different processing of these samples. And so we worked with them on this technology, and I was kind of their test case to get the robot to do the sample, the slide washing and all of the processing, which ended up being great because it automated the process. It makes it a lot simpler. It made that technology available to more labs in a very controlled manner. And so it was a very frustrating failure for a long time that turned into this kind of really neat area. I never would have put my bench equipment in a hermetically sealed box and tried to do an experiment if it wasn't something that we had to deal with the ozone gas.
That's fascinating. It's an incredible.
I say it now with a smile on my face. Let me tell you, in the middle of it, I was not smiling.
I'm sure you were shaking.
Everybody's got that moment in their PhD where they're just like, oh, I cannot figure this out. Stuff is going wrong. And everybody comes through it on the other side. So, please, if you're a PhD student listening to this, please remind yourself that it's going to be okay. You will figure out what's going wrong with the experiment, and it's going to be all right.
Do you feel like working through those sorts of problems prepared you for facing even more technical challenges in other parts of your career?
Yeah, I think it's a general sense of how you address problems, both from a scientific and an engineering perspective. And it's actually been very interesting. I was trained as a scientist. I grew up in scientific organizations. And then I transitioned to NASA, which is an engineering organization. And they're very different approaches to solving problems. They're related fields, but a scientist is kind of going to go out and explore and discover and do things differently every single time. That drives the engineers nuts. They need things to be repeatable exactly the same way, and they need deep test data with the same conditions. And so it's been really neat for me, coming from a science background, moving into this engineering organization and kind of understanding like how these different approaches to that same kind of general problem that we have of discovering things or building things or getting to a final product. And so, some of that some of that scientific work that I did early on with building this box and the ozone and the robotics ended up being a bit more of an engineering problem. And so I try to bring that back into scientific experiments and bring some of that engineering rigor. People are much better about lab notebooks these days. They've got these electronic systems, right? But back in the day, people were terrible about putting stuff in lab notebooks. And then when you come up with a problem, like some enzyme has gone bad and nobody's experiment is working, you don't have that documentation that you need. At the same time, sometimes the engineering work, it just gets way too drilled down into replicating the process that they've used to follow. And they lack that scientific creativity, that spark of like, let's just do something completely different and see if this wild idea works. And so it's been really fun for me to kind of straddle those two worlds and see the strengths and weaknesses in each of those and bring those together.
And you're still you're perhaps even more involved in robotics work now. How has like the evolution of robotics technology, how much has that changed over the course of your career?
Yeah, it has changed a lot, and I am involved in some of the robotics work that we're going to do, for example, on the surface of the moon, and then did a lot of robotics work when we were up on the space station. And so there we have really, it's a giant robotic arm that we use to capture incoming cargo vehicles. So for a cargo vehicle to have all of the systems on it to be able to dock to the space station, it needs things like LiDAR and retroreflectors and a whole control logic that it can come and find the docking port. So it's actually easier for us to use the robotic arm and have the cargo vehicle just kind of stay in position and we go grab it. And then we dock it to the space station. What that does is then there is just one control mechanism, which is the robotic arm, that brings the vehicle into the space station. And it's there all the time and we don't throw it away and it works. So that's very cost saving. For the cargo vehicles that come up, they don't necessarily have to put those systems in to control the docking. But then the burden falls on the astronauts now. We have to control the robotic arm. So we learn a lot about this system and how this has evolved. And we can certainly see as we're going to the moon and to deep space, the evolution of robotics there, computer vision, being able to take an image on a screen and have a lot more data versus just somebody with their eyes controlling an arm, making that a more automated process, and that ensuring that there's still some human supervision, but now the robot is doing a lot more of that work of understanding where something is in space and time and doing all that image processing.
Like a whole other help companion.
Yes.
So after you completed your studies at Stanford, was that when you went to the Whitehead Institute? Was it immediately following that? Take your time.
Yeah.
I need to ask longer questions.
Yes. So after I finished at Stanford, I had a pretty strong idea of what I wanted to do. I was doing a lot of the pox virus and genomic work. But we had been looking at it from the human immune side, and I wanted to continue that. But we really had been almost completely looking at the immune side of things. And I got very interested in digging into more about what the virus does when it gets into a cell. And I started to get very interested in what was going on with the genome of the virus, what it was producing, what genes it was producing. And for me, pox viruses was just that perfect genome size, right? It was enough genes, 200 genes or so, to be doing some really interesting things and have complicated programs of gene expression with multiple different phases. But it wasn't like a bacteria that I just felt that it had too many genes, and then also there's too many different kinds of bacteria. I was like, let's get it down to like I can fit 200 genes in my brain and have a really good understanding and know all their names and keep track of them and know what they're doing. So this was probably just like, my mind is not complex enough to actually go think about a bacteria. So a nice big virus was just the exact level. And then, of course, you have the complexity of the human immune system on the other side. So some of the things that I was interested in were mapping the program of gene expression in pox viruses. And it was a program that was pretty well known in terms of all of the pox virus genome had been completely sequenced. The classical virology techniques had been applied to understand more or less what these genes were doing and when the transcripts were turned on. And then I really wanted to look at it instead of following a single pox virus gene and understanding when that transcript was turned on and what the function was of that of that protein ultimately, I wanted to look at it from a genomics level and try to understand, okay, when the poxvirus is in the cell, kind of in real time, what's going on with all of these different genes as they're being turned on, and use the approaches that we had used to look at the human side of things, and then turn around and look at the virus side, with the idea being that we would bring those two things together. And so this was also kind of at the transition boundary from microarrays now to more sequencing. As the human genome came out, sequencing became a little bit cheaper and more reasonable. The machines were getting bigger. We could do sequencing faster. And so, I definitely could have gone and done all that as a postdoc, but I was having a hard time finding a lab that was really going to put those things together. And so I thought, well, one of my friends had participated in a program at UCSF where you come right out of your PhD and you're a fellow and you have a research lab and you have PI status, so you can apply for grant funding. And I thought, well, that's a long shot, but I'm going to try. There were 3 of these programs in the country, one at UCSF and then one at Harvard and one at the Whitehead Institute at MIT. So I moved to Boston without having a job. I just moved. I was like, well, 2 out of the 3 programs are there, and the odds are a little bit higher. So I moved to Boston and then continued a little bit of my graduate work after I had finished my PhD. I called up a friend at Harvard and said, hey, can I come squat in your lab? Do you have a little bit of a bench space that I can, I have some funding still on a grant that I was on at Stanford, so you don't have to buy me reagents or anything. I just need a little bit of bench space. He was a very nice guy and let me just have a little chunk of real estate in his lab. And then I applied for these fellows programs and got the fellowship at the Whitehead Institute and started my own lab there. And so it's like being a PI. You get lab space, and a little bit of funding to help you start, and then you can apply for grants. And so I started applying for grants and started building up the lab. And part of the piece that I wanted to do in terms of looking at the virus side was really explore what happens to primary samples. So when we talk about a virus in a lab and the virus infects the cell, we've got a viral culture, but that culture has been passaged many generations. So at some point we've taken an Mpox scab, right? And we've grown the Mpox virus out of that. And then we've passaged it multiple times. And every time you passage a virus and it goes through some life cycles, you could have mutations, you could have things change. And so I started getting really interested in clinical samples. Let's look at what's happening right at the point where you've got an infection. What's that virus doing? Maybe I don't trust these viruses that we've grown in the lab for a very long time. And with that focus on clinical, then you can capture all kinds of metadata. So you can capture medical record information about the patient who's donating the sample. You can try to understand what's going on with their immune system. And so it's a bit more of this direct clinical measurement of what's going on in the virus versus an in vitro experiment where I'm taking the virus out of the human, I'm taking human cells out of the human, and I'm mixing those together. At that point, it used to be called monkeypox. It's called Mpox now. It really only existed in a few places in the world, and one of those was Democratic Republic of Congo. And so as I was starting my lab, one of the things we really wanted to do was build up the lab to be able to do all those in vitro experiments, but we wanted to go and actually, number one, we wanted to treat patients and get patient samples. And so I worked a lot with USAMRIID to set up additional research facilities in the Democratic Republic of Congo. And so it was a great mission. They had a clinical side of it, so they were treating patients. They were treating patients for free. So we got to see that side. And then we had an IRB research protocol. And if the patients wanted to donate blood or donate a scab, they could do that, and we could study that for scientific purposes. And so for me, that was really neat. That was kind of the most clinical that I had gotten so far. And again, it was back to that patient interaction, in an incredibly challenging, resource-poor environment. And so, the clinic had a great success rate. And seeing people walk out of there alive, making sure that Mpox causes kind of whole body pustular rash with the particular clade of virus that we were studying, and that can get in the eyes. It can cause blindness. The pustules can get infected. You can be at risk for sepsis. You can have scarring if it's really getting infected. It can have a lot of problems with the skin. And so it was an incredibly visible disease to see. And, again, I wasn't part of the clinical side of things, but it was really great to watch what they were able to do over there treating patients with this pretty rare disease. One of the reasons we all wanted to understand it was because it's similar to smallpox, which is eradicated now. But of concern as a bioterror agent that this could come back again, and we're not vaccinated right now, and this could be definitely a problem if it ever got introduced as a bioterrorism agent. But then also we were concerned about mpox and potential for spread globally, and unfortunately we have now seen that in the subsequent years. We were really trying to do as much research as we could so that we felt like there was probably at some point going to be some outbreaks. Prevention is the key there, and prevention of transmission. But if you have transmission, what you really want is a therapeutic to treat the disease, and then you want a vaccine to prevent it from being transmitted further. And so we worked a lot to develop an animal model of smallpox and to take these clinical samples, and that provided some of the foundation for other researchers to come in and do their therapeutic development or their vaccine work development. And so it was nice to see, like a decade later, that these things are available, and that they're actually being used in the mpox outbreaks that we're seeing in current day.
That's incredible.
That was a very long answer.
No,
to your
that was
question.
amazing. That was amazing. Well, I think a follow-up question to that, and I do want to make sure we have enough time to get actually to your NASA experience too, but is how did, from sort of a public health perspective, being in that environment, in that different cultural environment, that different, clinical environment, what did you find? How did people who are patients and participants in these research studies, how did you find their understanding of the importance of, minimizing transmission, getting vaccinated? What was the public education process like while you were there?
Mm-hmm. Yeah, it was definitely, it's very different than an experience of working in a research lab in the United States. It's an incredibly impoverished area. There's not a lot of resources. The healthcare system is minimal, fragmented, and the country just doesn't have the resources to support public health as we understand it in the United States. One of the things I loved about that project is that we were very integrated with the country's research system and their public health system. So when we went out to the field, we didn't just come in with a bunch of American doctors. We actually had the Congolese doctors out there treating the patients. Like, number one, they had the best experience. They really knew what was going on. And then they also knew their patient population, and they knew what were the other things that we needed to think about in terms of treating the disease. They were the world's expert. They'd seen the most cases of disease. So we worked with the INRB, which is the National Research Institute in Kinshasa, and they had a very robust, they've got a mpox program, an Ebola program. And so we were sharing scientific knowledge back and forth with them. But it was an incredible challenge. I think anybody who's traveled outside the United States to a very poor area, it kind of resets your thinking about life and what we have, and just the unjust nature of the world that we live in. It helped a little bit to be there and to be doing something we felt like we're making people's lives better. But one of the things we really thought about was we don't want to drop in, parachute in, do some stuff and leave. This is the story of really what happens when Western countries go into Africa, and it can have a lot of really bad consequences. So part of the program was actually getting equipment in to make sure that the area that we were at had a diagnostic lab that they could continue to diagnose. We wanted to make sure that we trained local people to be lab technicians, so that the idea is that they're then running their own lab. We can step back from that. And then the public health piece, education and outreach to the community. And they're not going to listen to somebody like me. Like, this is like with the HIV thing, right? Like, the kids are probably not going to listen to the adults that are giving them some boring lecture. But the integration with the local public health system and making sure that we were working to help people understand what caused this disease, how to recognize the symptoms, that if somebody in their village had it, that this is what it's going to look like, that we have this clinic that's providing care for free. Bring the patient to this clinic, and we're going to help them, and not just help that patient, but help their family members who might also be exposed, and really provide that supportive level of care to the entire family that's bringing the patient in. And so that was important. And I think, it's working across these different cultures, like there's a lot of potential for misunderstanding. We're not even speaking the same language. We were both speaking French in cases where we could, because it was second language for, my French is terrible, and like their French was much better, but nobody's speaking in their native language. And we have really different perspectives on where we're from. And so I think what did the most amount to kind of bridge that divide was showing up and doing patient care. And people saw, hey, they're not only is the patient being cared for, their family's being cared for, and they're walking out of there and they're doing much better than they would have otherwise. And that, that spreads, and that word spread, and we got a very positive reception. And I think that's incredibly important. We're just, the U.S. is so bad about going in and having our worldview and imposing it somewhere. The best thing was to come in and to listen to what that community needed. Like we came in and the hospital didn't have beds. And so before we started the lab work, we built beds out of trees. It's just be in that place a little bit and understand that environment was incredibly important to us. And that it really changes your worldview to live in a place like that. We would kind of go back and forth. I'd have to be back at my lab at MIT and then go back out there. But we were always in the same place. We were interacting with the same people and kind of getting this lab set up. It's profoundly changing. I think I've carried that with me.
That's such important work, and I really appreciate your perspective on U.S. and how we approach other countries in the world. At what point did you mentioned earlier that you had sort of let go of some of the astronaut dreams early on, or had focused on other things because you couldn't get a PhD in astronauts. When did you start to consider, leaving the lab at Whitehead and applying for NASA or applying to be an astronaut? Was that something that you had sort of mapped out in advance, or was there a moment at which it came back in a rush and you decided now's the moment to do it?
Yeah, I was here at NIH a few years ago, and I told Francis Collins that it's actually all his fault, because the way this came about is I was working with one of my really close collaborators applying for an NIH grant. And anybody who's applied for an NIH grant takes a lot of work, a lot of sleepless nights, getting your data together and putting the documentation together. And so we were towards the end of the grant, but kind of struggling on one section, and we're talking over the phone and typing on our computers. And my friend Lisa said, we need a break. This is not going well. Neither one of us are thinking straight. And she's like, I was looking at other jobs online. And she said, I've looked at usajobs.gov, and there's an application to be an astronaut online. Isn't this ridiculous? And I laughed, and I was like, that's the funniest thing I've ever heard of, that there's just send in your resume to be an astronaut, which is actually how we recruit and select astronauts. And she said, we should apply to be astronauts instead of writing this grant. And I took her a little bit too seriously. So I applied to be an astronaut that night instead of working on my grant. And I didn't think that NASA would hire a biologist. I really thought this was this was a nice distraction from working on the grant. But as it turns out, we do a lot of biological research on the space station. Biology is, human biology is incredibly important as we're thinking about going out into deep space. And so it's not just all rocket scientists and engineers and physicists that are working at NASA. And so I put in my application, and I just really didn't think anything was going to come to fruition. But, my 5-year-old self was kind of excited that I'd applied to be an astronaut. And so I was very surprised a few months later when my references called me and said, I've been contacted by NASA. They want me to write a letter about you being an astronaut. And I was trying to figure out if Lisa had somehow pulled off the most massive prank in history. But it was legitimate. So I went down to NASA and I started the interview process. It was just these, these moments in your life that you're like, there's one tiny little thing, one phone conversation on one day that changed sort of what I want to do and what I want to do in my career. But I'm very grateful for it. I still show up to work every day thinking like, I don't know how in the world they selected me. I better do a really good job here because there are so many talented people that I interviewed with. And so it was a completely different direction for my life and has led me to different science and science in places I never thought I would be.
I do love that the bureaucracy of NIH is what pushed you into space. The point in our favor, truly. So how and of course you're very well known for having been the first person to sequence DNA in space. What drove you to want to do that? So what was your experimental passion for doing that? What did you want to learn? And also, how did you physically do that? Because it seems very difficult to do.
Yeah, we got interested. So this wasn't just my experiment. This was actually a huge project that was working on this. Really, the driving factor was microbiology. So on the space station, if there's some green fuzz growing on the walls or something in the water system, or an astronaut's come down with an infection, the way we would previously diagnose it was we would take a sample. We'd streak a plate and we'd have to wait until a cargo vehicle was undocking from the space station. And this only happens every 3 months or so. So you would have to wait potentially up to 3 months, streak the plate, wrap it in parafilm, put it on a vehicle that's going to land back down on Earth. And at that point, it was only Soyuz that was landing on Earth from the space station. So it would land in Kazakhstan. NASA would have to send over an airplane to pick these samples up, along with other things that they were picking up, and bring it back to the lab at Johnson Space Center, and then do our standard microbiology. And so you can imagine, sepsis moves a lot faster than the speed of that diagnostic process. And so we were concerned. This was a risk for NASA. We want to know about environmental exposures because we are living inside our laboratory on the space station. It's completely enclosed, of course. The air is recycled, the water is recycled, but this means that any environmental toxin or exposure, because you are living there, the astronauts would be exposed to that. And so we're worried about pathogenic bacteria, yeast, all of these kinds of things, as well as human infection. And there's a cool experiment going on right now to try to figure out if we've got antibiotic resistance on the space station. So that might terrify some people when we get the results. But it was these plates, and the lab was very far away from the point where we needed to sample. So sequencing allows us to do that in real time. And the idea behind this experiment was if we could get sequencing to work, we could take a swab, prepare our DNA libraries, and then sequence it. And then we have a connection back to Earth. We've got either could send that Earth data back to Earth to be processed, or we could bring a computer on board and we could do the processing ourselves, and we could identify all the microorganisms in that sample. And the idea being that we could probably speed this process up to less than a week instead of 3 months. And the first part of it was, can we even sequence in space? And so NASA, usually when they do an experiment, it's very involved. It's got a lot of engineering checks. It does make NIH look like the easiest bureaucracy in the world. So you guys don't worry about those R01s, because if you're going to send an experiment to space, there's a lot of paperwork to do, which makes sense. You have to figure out the safety of it because the astronauts are brushing their teeth next to it. And so that was kind of holding back scientific research a little bit. The other thing was that they wanted it to work. And so you had to have so many preliminary data that it would take years and years before you were allowed to fly that experiment. And that's not a good way to do science. That's not how it works. So they did a program that they said, all right, we're going to take more risk, not on the safety side, but we're going to take more risk that these experiments don't work. And if they don't work, we're going to figure out how they broke and we're going to fly them again. And so the sequencer was one of the first few experiments through this program. And so we said, we really don't know if this is going to work at all, but we're not going to waste a whole bunch of time on Earth trying to figure that out. We're just going to fly the thing. And if if it doesn't work, we're going to figure out where did it break, what broke, what part of the process do we need to go address. And so we fully thought that we would probably be sending a few more sequencers and getting to our answer later. And so some of the things that we were thinking about were bubbles. So in microgravity on the space station, everything is in free fall. You're in free fall around the Earth, which means that you can kind of think there is no gravity, right? People say 0 g. It's really very minute amount of gravity. What that does is that if you've got bubbles in a tube on Earth, the air eventually goes to the top and the bubbles are up there and then they pop and the liquid goes to the bottom. You get this automatic phase separation just due to the fact that we're on the planet and we have gravity available to us. That does not happen in space. So once that bubble gets in that solution, that thing is going to stay there. And it's going to, find its friends and coalesce sometimes, or it's going to break into smaller bubbles. But unless you've got motive force and a gas trap, you're stuck with that bubble. This is very bad for sequencing. If you get an air bubble that sits on top of, takes over a whole bunch of nanopores, that area of the chip is going to be dead. We're very worried about bubbles. We were worried about data. How is that going to interact with the data system? And so we had to work a lot to work on a separate payload LAN, and we just had a separate tablet that was connected directly to the sequencer so that we didn't interface with any critical station systems. There's some other concerns in terms of thinking about thermal. We couldn't have a piece of equipment that produced a lot of heat. Otherwise, you have to weld it onto a cold plate because there's not buoyancy-driven convection in space. And so if something generates heat, it is just going to generate heat and it's going to stay right next to that equipment. There's not, you can direct a fan on it, but air cooling is not very efficient. So you usually have to stick it onto a cold plate and do liquid cooling. And then that has to hook into the space station cooling system. So the paperwork stack theoretically is getting higher and higher. And so we did find the technology that was going to work well, which was the nanopore sequencing. It's a small instrument, doesn't generate much heat. It could connect to a small tablet computer next to it. And then it was just really up to me to be very diligent about not introducing any bubbles into the sample. And we kind of did our whole preparation steps to try that. And the good news story is, for the first time in my life, an experiment worked the first time. If you're going to have one experiment that just works flawlessly out of the box, it's good to have the one that like 1,000 people are watching on the ground to make sure that you don't screw up. It is, I call it the single most nervous pipette that I've ever had. One pipette to load the flow cell, and there's people watching, and I'm like, wow, Kate, just don't mess this one up. Just don't send your very carefully prepared library in a bubble of liquid floating through the space station, or put bubbles in the nanopore sequencer. There were a lot of things that could go wrong. So I kind of got in my Zen place and I pipetted very carefully. And so that worked. And we published a paper. We were able to reassemble. We sent up a mix of genomes. It was E. coli and lambda and mouse. We were able to reassemble E. coli genome out of that mix. And half the samples were still on Earth, so we could actually get ground truth and determine that sequencing worked just as well in space.
Maybe for the next 5 minutes, our final question can be, in terms, like, because I know you decided you would join the U.S. Army Reserve, and you're looking at training for the Artemis mission, or you are actively training for the Artemis mission. I guess what do you see as your next steps in your astronaut career, and how does that dovetail with your biology research that you're currently doing?
Yeah, I feel like I'm doing a lot more biology research these days. There's a period where you're just training to be an astronaut. But now that I've flown twice, I still have to spend a lot of time doing the astronaut training, which I can't complain about because, we fly jets and we test spacesuits underwater. So that part is all good. But we're doing a lot, some of the science work that we're interested in right now. So I'm super interested in, eventually, when we go to Mars, we're going to go to Mars to find life. Like, this is one of our major objectives. Unfortunately, as humans and all of our associated microbes and our equipment, we bring a lot of life with us. And so this is kind of a terrifying situation in terms of negative controls. We're trying to detect something that may or may not be there in minuscule amounts with the biggest source of contamination you could possibly think of kind of walking around. Now, we aren't walking around, in our lab coats on the surface of Mars. Of course, we'll be in a spacesuit, but we can't really process the spacesuit in a sterile facility. There's been a lot of work over the years to sterilize spacecraft before we send them out into the universe. And there's a whole field of this called planetary protection. And we have a planetary protection officer at NASA, which I think that's got to be the coolest job title in the world. And they do a lot of work about thinking about this. And so I was thinking about when we go to the lunar surface, we know that the lunar surface is sterile. We know that there was no life on the moon. And so if this could be the world's most massive experiment for life identification, if we go and we're collecting samples from the soil, from the regolith, if we're collecting swabs from the suit. So my idea is that we build this kind of spatial temporal map of the humans exit the vehicle onto this sterile planetary body surface, and we take samples and then we sequence because we want to see what's the persistence of DNA. So probably most of the organisms that we bring aren't going to be alive for too long, but it's very interesting to see that, how long are they alive? But then, as they get killed, they degrade, but they still leave this molecular residue. So we want to look at DNA. We also want to look at proteins via mass spec. I'm going to put the disclaimer that this is not a NASA-funded experiment currently. This is Kate's favorite experiment, which I just submitted an abstract for, and I just keep talking about it. So I think it's so cool, like at some point we're going to decide that we want to do this to prepare for Mars. And so this idea would be that we would sequence these samples as we go out in space and time and try to determine the extent of the bioburden. So who's there? Like obviously human cells, but which bacteria are there? How do they degrade in different ways? Like human cells are going to degrade in a different way than a bacteria, gram-positive versus gram-negative are going to degrade differently. How does that affect the molecular residue? What is the point where we can no longer detect that molecular residue if we get there on the surface of the moon? And this is going to just kind of map our contaminated zone that we're going to bring with us to Mars. I think that will inform future efforts to figure out how to reduce the bioburden. How do we reduce some of this contamination so that we're not bringing it to Mars? And it's the coolest experiment because it's like the first time that we're going to map a biological ecosystem in a completely sterile world. I figure we've got one shot at this. Like we need to do this. We need to do it right? Because, we're going to land on different places, the moon, and this ecosystem we bring with us is not going to get transmitted. So kind of every time you land on the surface of the moon, you're going into your sterile area. But at some point when we have more equipment and more vehicles in the moon, we won't be guaranteed the best negative control in the galaxy. This is probably what I should put as my grant title. So I'm really excited about that. And, when we get back to it, I can probably explain a lot more about synthetic biology and some of the other things that we're doing.
Is that grant application going to NIH or NASA?
It's not even a grant yet. I'm just submitting as an abstract to a conference to start talking to people about it. Well, maybe I should write an NIH grant proposal.
You probably should. You're about to talk to a lot of people who would give you good advice about where to submit it. Would you take a really good shower before you do the moon example, or would you take a bad shower so that you would have more microorganisms on you?
I think we'd want to take the best. We can't even take a shower in space, but we would do kind of the best cleaning we could of ourselves and the spacesuit because we want to simulate what it's going to be like at Mars. So we're going to probably use all the equipment that's available to us that's authorized to clean the spacesuit and the airlock and kind of get it as good as we can. And then we just want to go see. We want to quantify the problem, like how much is there? I'm always interested in these combined microbial communities that have a bunch of different microbes in them, like who's there? What are they doing? How long do they last? Which microbes essentially have the stuff that it takes to leave the most amount of molecular residue, because that's what we really want to make sure we get rid of.